So you've learned about how a neural signal travels along a single neuron. The question now becomes, how does one neuron communicate with another adjacent neuron? As you recall, there is typically a gap between adjacent neurons called the synapse. And so you cannot have an electrical signal travel from one neuron to the other across that gap. It would be like disconnecting the wires between a light bulb and a battery. Your current won't flow. So the human nervous system has a way to overcome that problem. Specifically, chemicals called neurotransmitters are released into the synapse or gap by one neuron, and then they travel passively across the synapse, possibly to be picked up by another adjacent neuron. For the most part, this is how one neuron communicates with another neuron in your nervous system. But we do need to look at these processes in a little more detail. We're talking about how one neuron communicates with another adjacent neuron. So I've presented two neurons at the top of the screen. Neuron number 1, and of course you know its parts, is the sender neuron, and neuron number 2 is the receiving neuron. I've also shown that there's a tiny gap between those two neurons. So I'm showing the synapse. At the synapse, I've drawn a red box and then a larger red box to show a magnified view of events at the synapse. When you look at the large red box, you see that at the top is the presynaptic terminal, and I've drawn the postsynaptic dendrite in yellow at the bottom. Postsynaptic is the opposite of presynaptic, and postsynaptic means after the synapse. So here's what I want you to know about synaptic or interneuronal transmission. Imagine that a signal has traveled along neuron number 1, our sender neuron, and it has reached the presynaptic terminal. In the presynaptic terminal, there are little structures called vesicles, and a vesicle is a container. That's all it is. And those vesicles in the presynaptic terminal contain chemicals called neurotransmitters, NTs. When that neural signal reaches the presynaptic terminal, what happens is that those vesicles will move over to the cell membrane, the outer covering of the neuron, and they will merge with the cell membrane and release their contents into the synapse. So now you have neurotransmitter just floating passively around the synapse. Some of it will reach another neuron. Here it's neuron number 2, the receiver, and that neurotransmitter substance will bind with receptors on the dendrites of neuron number 2. Again, some of the neurotransmitter will reach the receiving neuron, the adjacent neuron, and bind with receptors on the dendrites of neuron number 2. If enough neurotransmitter reaches neuron number 2 and binds with receptors on the dendrites of neuron number 2, then a signal will travel along the length of our receiving neuron as well. You definitely need to know that process, but that's not quite the whole story. Reuptake is also important. And by reuptake, I mean that there are enzymes in the synapse that break down unused neurotransmitter, and then the parts can be used to synthesize new neurotransmitter, store it in the vesicles, and our sending neuron can do all of this again. Some people refer to reuptake as a cleanup process. You can think of it as cleaning up unused neurotransmitter from the synapse. And this is absolutely essential for our survival. A neuron is not going to be functioning properly if it can only send one signal across the synapse to another neuron. It needs to be able to do this repeatedly. So there you have it. That is what I mean when I talk about inter means between, interneuronal transmission or synaptic transmission. Basically, a neural signal travels along neuron number 1. It reaches the presynaptic terminal where there are vesicles containing neurotransmitters. The neurotransmitters are released into the synapse. Neuron number 2 picks those up, and the neurotransmitter binds with receptors on the dendrites of neuron number 2, the receiving neuron. And then there's also reuptake, or a cleanup process, so that unused neurotransmitter can be removed from the synapse, broken down to its components, and the cells can synthesize new neurotransmitter substance, store it in the vesicles, and start all over again. The reason this is important is because it allows us to understand the effects of many types of drugs on behavior. For instance, consider the drug curare. It is a drug that has been used by South American Indians. They've extracted it from the leaves of plants and then dipped their arrowheads into it in order to increase their hunting success. Today we know what curare does. It binds with receptors on the dendrites at the neuromuscular junction. Now that's a big word and I won't test you on it, but look at it. Neuron, muscular, muscles, and a junction is connection. So what curare does is it binds with the receptors at the neuromuscular junction, blocking the normal neurotransmitter that is involved in those neurological processes. That neurotransmitter, you don't have to know, but it happens to be a neurotransmitter called acetylcholine. Think about it. If the receptors are essentially blocked, then the normal neurotransmitter cannot have its effect. And so the result of curare is paralysis and perhaps death if the muscles of the heart and lungs are paralyzed. The result is, if you are a South American Indian, that your hunting is going to be far more successful. Because if the drug gets into the bloodstream of a prey animal, then it will be paralyzed and more easily caught. Let me share a little bit of lab lore with you about the effects of curare. This was told to me when I was a student. I've never found a source where the people are identified, but it's a useful illustration. This is what I was told, that a scientist wanted to experience the effects of curare. So he had to enlist the assistance of some friends in a laboratory setting, and curare will result in paralysis, of course. So they had to maintain him on a life support system while he was experiencing the effects of the drug. After the effects wore off, he was able to talk about what he experienced, and what he said was that while he was under the effects of curare, he heard everything said in the room, every sound. He felt every touch on his body. He just could not move. He was paralyzed. This is a drug that affects the neuromuscular junction, where the neurons control the movements of muscles. It has no effect on incoming sensory information. Therefore, curare should never be used as an anesthetic. It does not do anything to stop incoming sensory information or to stop pain. What it does is stop any expression of that pain, any muscle movements.